Hi everybody. Okay, so today I'm going to teach you about something called a gel. And at the end of the day, a gel is a molecular biology technique that separates large pieces of DNA from small pieces of DNA. There's a lot more to it than that. It's not terribly crazy, but this is the thing to remember. Large pieces of DNA get separated from small pieces of DNA. The gel technique has actually some fancy names. Sometimes it's called electrophoresis. Sometimes it's called agarose electrophoresis. Sometimes if you use something slightly different, it's called polyacrylamide gel electrophoresis. All are really the same in concept though. Okay. So like I said, what we're going to do here is separate DNA by size. And that's based off of a really simple principle. I just happen to have a model of DNA here, and if you look at it, around the outside there is red and yellow phosphate groups. These phosphate groups here have a negative charge, and all I'm going to do is apply a positive charge somewhere away from the DNA, and that is going to pull the DNA towards the positive charge. Now, I'm going to put a little bit of an obstacle in between the positive charge and the DNA so that smaller pieces of DNA travel faster through the obstacle to the positive charge. You can think of it in kind of metaphorical terms. Imagine a forest with candy on one side and kids on the other. Kids that are holding, kids in pairs or two kids holding hands will travel much faster through the forest than 17 kids holding hands trying to get through the forest. 17 kids are going to get caught up in the trees while the two kids are going to be able to maneuver fast through it. So the thing to remember is small pieces of DNA are going to travel very quickly through our forest or matrix. Cool, so what does this look like? Well, this is our basic contraption. There's a lot of different versions of them, but they all work on the same idea. Basically, I have two electrodes. I'm going to apply a positive charge on one side, a negative charge on the inside. Inside this chamber, I'm going to have this little gel and a tray. So this is a tray. This is the gel inside of the tray. It's basically the same consistency of gelatin, but it's actually made of a sugar called agarose. What I'm going to do is put DNA in small holes that I've made inside the agarose in the gel. The holes are right up here. And then I'm going to fill this whole contraption up with water, or actually salt water, called usually TAE or TBE. I'm going to apply the positive charge here. The DNA is going to run through the whole thing, and the smaller pieces are going to end up closer to the positive charge down here, and the larger pieces are going to end up here. I'm going to add a little bit of a DNA dye here so that the DNA glows, and I can really see the DNA. So let's say I have a sample I don't know the size of. I would put it over here. And then I would put maybe sizes of DNA that I knew. Maybe I'd put a small piece like with 10 base pairs, 100 base pairs, and 1,000 base pairs on this side. And then I would run those and see how far my sample ran in comparison to them. And that's basically it. Again, the basic concept here: DNA, negatively charged, positive electrode, forest. Small pieces of DNA are going to move quickly through this thing. Bigger pieces are going to get stuck up top. I need to make sure that I dye my DNA so that I can see it. And then I'm going to need to make sure that I put some kind of standard in there, standard sizes of DNA, so I can compare my sample to it. Thanks. Yeah.